Five, 4, 3, 2, 1, 0. All engine running. Liftoff. We have a liftoff.
Ladies and gentlemen, thank you once again for tuning in to Live with Andy. I'm your host, Andy Dybala. How do you like the added sound effect, ladies and gentlemen? I'm learning. I'm learning the roadcaster buttons a little bit here as we go along, and you can see summer is pretty much here in West Texas. I wouldn't call it the dog days just yet, but it's been 95 degrees 2 days in a row now, so we don't got the AC yet on inside, and I had to ditch the cowboy yet, so I got a cowboy hat, so I got my little Sons of Anarchy look going on right now. I don't have the motorcycle in the garage per se, but I do have a nice little Honda scooter that I take around town. But today I actually have a little different show going on. As you guys know, I've had, I mean, some of you guys know that I've had over 20, 30 different YouTube channels. A lot of my work has been taken down because it's been deleted, and just it's gone forever now. My catalogs, most of them got lost during the move from Neighborhood News Studio, Michigan to Atlanta to Texas. So I'm gonna redo a different show that I once did. You guys know I like doing my caveman sciences here, so I'm going to be digging into the origins, not of SARS-CoV-2. You guys know that I've done a lot of work into Ralph Baric and his genetic engineering of the SARS-CoV-2 virus, but I'm going to be digging into some of his work and some of his predecessors' work into the original SARS virus. If for those of you don't know, like the coronavirus laboratory, like the worldwide coronavirus laboratory, is in North Carolina, and Ralph Baric is the head of that now. But once upon a time, he was doing some coronavirus work and before he got to North Carolina, and there was questions when the original coronavirus came about in 2003 in China. There's questions then. Is this thing genetically engineered? They said it popped up in nature. They kind of blame like the wet markets. They were murdering civets in cages by droves in 2003 in China to try to eliminate the original SARS coronavirus. And a lot of people, scientists on the scene, were like, we've never seen this thing before in nature. It's kind of weird that it's starting to pop up. So I got a couple different articles I'm going to be reading here today. And I apologize. I am saying a lot of uhs and some stuff right now. I'm warming up a little bit. For those of you guys that don't know, I've been in the hospital. Before that, I was on a little bit of a vacation. So it's been a while since I've been in front of the camera and behind the microphone, showcasing some research. So bear with me for a little bit as I get warmed up and get into gear here. It takes a couple of shows. It takes a couple of days sometimes to get back in the groove. And thank you to all of you who have stuck by. Let me say hello to Black Rabbit, not the White Rabbit. Follow the Black Rabbit. Hello, how you doing? Thanks for joining. We got Deborah Johnson. She loves Caveman Science. I hope all of you love Caveman Science as much as I do. I have invited an actual scientists on the show. We're trying to work some things out so we can break some things down a little bit more intellectually than what I do here on the show. I'm a researcher. I dig into the articles, I dig into the science, but some of the times I don't know the little intricacies of things. So you got to have interviews every once in a while, to enlighten ourselves. And I'm going to be trying to do that a little bit more moving forward here on Live with Andy. So we are 5 minutes in, and let's get to the articles here. So this first one, this was actually from 1999. So we are going to be rewinding the clock here a little bit on Live with Andy on this Monday afternoon. Like I said, we're going to be digging into the genetic origins, the genetic engineering and the origins of the original SARS coronavirus. See, my tongue twisters. I gotta get used to talking on the microphone again. So this article is titled, "Is it possible to engineer viruses to perform specific tasks within a human host? That is, to create viruses that are beneficial rather than detrimental to human health." And shortly after this article is introduced, there are some problems start emerging on the scene. So this was in Scientific American, and it was answered by Leslie Snyder, a biotech. You know how you can ask questions to the magazine. So this is Leslie Snyder, a biotechnology instructor at Miracosta College in Oceanside, California, responded to the virus engineering question. Can we engineer viruses to be good rather than bad to human health? Greetings, Freedom123. Thank you. Thank you. Don't forget to like and share. Here we go. Not only is it possible to engineer viruses to be beneficial, it already is being done. Viruses have primarily been engineered for use by humans as so-called recombinant or subunit vaccines, and this is what gets to be important here in Ralph Baric's and our coronavirus work here as we go continue on in the show, these recombinant viruses. In this technology, the genetic material of a harmful virus is analyzed to identify the gene or genes that encode the antigens, identifying proteins that trigger the body's immune response. These genes can then be isolated and inserted into host cells, usually bacteria or yeast. The host cells, I need to turn my brightness up here. I can't see. It's part of my problem. The host cells in turn manufacture the antigens in large quantities. The antigens are injected into humans and result in the formation of memory B cells, which are tailored to the alien antigen, thereby protecting against the disease caused by the whole virus. The first recombinant vaccine designed for human use immunizes against hepatitis B. It was approved by the FDA in 1986. Viruses also have long been used as cloning vectors, tools for inserting genetic material into a variety of host cells. And this is where SARS comes into play, including bacteria, yeast, and mammalian cells. These host cells are grown in the laboratory under controlled conditions. Now, they're grown in the laboratory, these cloning cells. And this is something Ralph Baric and the boys and some gals were working on in the late 90s and early 2000s when it comes to different types of coronaviruses. So I think they were engineering the original SARS virus in the lab. So when you question like, hey, is SARS-CoV-2 genetically engineered? Is it lab made? It's like, yeah, the original one was. So the second one has to be as well, according to Caveman Science with Andy Dybala. There are many research projects that focus on the use of viruses to insert normal, healthy human genes into human cells grown in the lab. So they're using viruses for genetic engineering, pretty much, of human cells. Great. That's why I say like the virus, the actual coronavirus is genetically engineered, and it's and like the mRNA vaccine is almost the same thing. It's like a virus encapsulated mRNA strand that gets into your cell. There are many. Sorry, I'm trying to pick up where I left off. There are many research projects that focus on the use of viruses to insert normal, healthy human genes into human cells grown in the lab. Some of these engineered cells may be suitable for therapeutic use to alleviate the symptoms of disease or perhaps even provide a kind of cure. Such treatments are currently being tested for the treatment of emphysema and cystic fibrosis. These diseases affect lung tissue, and it is relatively easy to deliver the engineered cells to the lungs via aerosol sprays. So they can, they can deliver these genetically engineered viruses and cells to the lungs via aerosol sprays. Is that how they spread the SARS-CoV-2? Is it? The use of viruses to inject genetic information into cells of living organisms, for instance, to insert normal genes into the cells of a patient whose disease is caused by a genetic flaw, is much more difficult and will require a great deal of additional research. Nevertheless, researchers are pursuing a number of promising possibilities towards this goal. For example, some scientists are working on schemes to use viruses to deliver normal genes directly into the cells in the lungs of people who have cystic fibrosis, or into the cells lining the walls of blood vessels in order to prevent atherosclerosis. And I'm not 100% sure what that is, but there you go. They are using viruses to genetically engineer human cells. Now that is the end of the question, or the response by Leslie Snyder. Unfortunately, we're not going to be able to. She doesn't dig a little bit too much more into the science, so you're going to have to rely on me from this point on. Let me say hello to Rumble real quick. I've been doing my Rumble streams lately, and to tell you the truth, my Rumble streams have been picking up more steam than my YouTube streams. I've been getting more viewers on Rumble. So thank you, Rumble, for all the support, and thank you everyone for um following through at Rumble, because we all know YouTube kind of put an end to free speech on the internet at certain points, and they're banning everyone left and right still. So we got to have spaces like Rumble, safe spaces. I used to say, I always used to shit on safe spaces, but Rumble's mine. Rumble is ours. I like the camera. I like the hair. I think my hair's on point this afternoon, ladies and gentlemen. It's officially long enough where I think I might be able to ditch the cowboy hat, shave my beard, you know. That's what I was going for. Not many of you people know it out there. I was in a intermediate stage when it came to hair growth here. So, I've been saying, that the original SARS is might be genetically engineered. Now there have been scientists and biologists who were agreeing with that statement in the early to mid 2000s, and they wrote a couple articles on it. And this Dr. May Wan Ho happened to be one of these scientists, and I'm gonna read a little bit of her work here: bioterrorism and SARS, and it looks into if the original SARS virus genetically engineered. Now I'm gonna skip ahead here. I think I'm gonna skip ahead here to genetic engineering super viruses. So I'm gonna actually post this in the chat. If you guys want to read this, it's in the chat. You're welcome. Rumble and YouTube. I actually posted something in the chat for once. So here we go. Genetic engineering superviruses. This is from Dr. Maywan Ho. While the epidemic, and this is talking about the original SARS virus, while the epidemic has still run its course, a report appeared in the Journal of Virology describing a method for introducing desired mutations into coronavirus in order to create new viruses. So, this is something we know Ralph Baric was working on in his lab. This is what reverse engineering is. He was coming up with mutations in his computer algorithm, and him and Bat Lady in Wuhan were using those mutations and actually like creating spikes and creating different viruses in the lab to create vaccines. And I have some patents and some of his research work, some of his documents filed away on my computer here, and I'll be showcasing those at the end of the show if we have some time. But here we go. While the epidemic has still run its course, a report appeared in the Journal of Virology describing a method for introducing desired mutations into coronavirus in order to create new viruses. A key feature of the procedure is to make interspecific chimera recombinant viruses. It involves replacing part of the spike protein gene in the feline infectious peritonitis virus, FIPV, which causes invariably fatal infections in cats, with that of the mouse hepatitis virus. The recombinant M mouse feline virus will no longer infect cat cells, but will infect mouse cells instead and multiply rapidly in them. Now, I do have this work that was done in the laboratory. I do have the scientific research. I'll be showcasing you in a little bit here. These and other experiments in manipulating viral genomes are now routine. It shows how easy it is to create new viruses that jump host species in the laboratory. In the course of apparently legitimate experiments in genetic engineering, similar experience could be happening in nature when no one is looking, as the SARS and many other epidemics amply demonstrate. It is not even necessary to intentionally create lethal viruses if one so wishes. It is actually much faster and much more effective to let random recombination and mutation take place in the test tube using a technique called molecular breeding. Millions of recombinants can be generated in a matter of minutes. These can be screened for improved function in the case of enzymes or increased virulence in the case of viruses and bacteria. In other words, geneticists can now greatly speed up evolution in the laboratory to create viruses and bacteria that have never existed in all the billions of years of evolution on work. Now, this is pretty much what Ralph Baric was doing in the lab in North Carolina. He was creating new SARS coronaviruses that never before existed on Earth. He's cooperating with the lab in Wuhan. Lo and behold, there's an outbreak, and he happened to have the remdesivir, the remdesivir cure on deck that was created in the same lab. It was used as one of the first therapeutics. But here we'll continue with the article here as I stumble along reading. Sheesh. Controlling bioterrorism. John Steinbrunner, University of Maryland arms control expert, has been calling for mandatory international oversight of inherently dangerous areas of biomedical research, specifically an international body of scientists and public representatives to authorize such research. I don't think I want to read this part. Blah, blah, blah. Who needs bioterrorists when we've got genetic engineers? But what caught the attention of mainstream media was a report in January 2001 of how researchers in Australia accidentally created a deadly virus that killed all its victims in the course of manipulating a harmless virus. Disaster in the making. An engineered mouse virus leaves us one step away from the ultimate bio weapon, was a headline in the New Scientist article. The editorial showed even less restraint. The genie is out. Biotech has just sprung a nasty surprise. Next time, it could be catastrophic. Now they created a like new mouse pox virus in the laboratory, and they were testing it on the mice, and it was just killing all of them pretty much. So they're like, yeah, we got to put this thing under wraps, and they hit all the scientific work in Australia because they were under the microscope, literally. The SARS episode should serve. Oop. Let me try that again. They were under the microscope, literally. The SARS episode should serve as a reminder of some simple facts about genetic engineering. In the first place, genetic engineering involves the rampant recombination of genetic material from widely diverse sources that would otherwise have very little opportunity to mix and recombine in nature. And as said earlier, some newer techniques will create in the matter of minutes millions of new recombinants in the laboratory that have never existed in billions of years of evolution. In the second place, disease causing viruses and bacteria and their genetic material are the predominant materials and tools of genetic engineering as much as the intentional creation of bioweapons. And finally, the artificial constructs created by genetic engineering are designed to cross species barriers and to jump into genomes, i.e., to further enhance and speed up horizontal gene transfer and recombination. Now acknowledged to be the major route to creating new disease agents, possibly much more important than point mutations, which change isolated bases in the DNA. With genetic engineered constructs and organisms routinely released into the environment, we hardly need the help of terrorists. That may be why we are coming up against new epidemics of viral and bacterial diseases with increasing regularity. Mother Nature is not the ultimate terrorist. We are. And this was written in the early 2000s, and Dr. May Wen Ho has been right. You know, you've seen like Zika virus, Ebola virus, Marburg virus, all these coronavirus, all these different viruses and like different flus. You got bird flus and all this stuff breaking out. We got pox viruses breaking out again. And all this stuff is being genetically manipulated in the laboratories. And I here think that's what happened with the original SARS virus and SARS-CoV-2. And most of that work was done with Ralph Baric and coronaviruses. So I have another, a bunch actually more articles here. And let's get into them a little bit. Once again, ladies and gentlemen, this is Caveman Science, so bear with me when it comes to some of the sciencey aspects of these things. You know, I'm just your average Joe digging into some of this stuff, trying to break it down for my average Joe audience, my caveman audience. At least we're not grunting. So this article. This is actually some work that Ralph Baric cited in some of his SARS coronavirus research. Reverse genetics of the largest RNA viruses. So they're trying to figure out how to reverse engineer viruses. And coincidentally, the coronavirus is selected because it's the largest virus in the family, making it the easiest one to work on. If you want to add different like endo or ectodomains, if you want to put different spikes on the virus and you want like a larger area to work with. So the coronavirus is chosen. I'm not going to um, read this article, but that's pretty much what it gets into. And because it's the largest virus, they're able to use it to reverse engineer into making different recombinants that never before existed in nature, stated by Dr. Mei Wan Ho. Now, this has all taken place in the late 90s and early 2000s, even before the original SARS came out. So we got retargeting of coronavirus by substitution of the spike glycoprotein ectodomain, crossing the host cell species barrier. Now, this was written in, when was it? Received in July 1999. So this is them creating pretty much different coronaviruses in a laboratory in the late 90s. Right here. So when Dr. Mei Wan Ho says that the original SARS virus is a product of genetic engineering, I think she's actually correct. So we got the retargeting of. I mean, I'm going to read a little bit of these, just the abstracts. I got a couple different abstracts I'm going to get into. Retargeting of coronavirus by substitution of the spike glycoprotein ectodomain, crossing the host cell species barrier. So, like she said, you want to be able to cross the host cell species barrier so it can infect humans or it can infect civets or it can infect mice. And they were using the spikes on the outside to do this. You know, that's what floating around in your system, the spike attaches to it, you know, then kind of absorbs the cell and it takes the mRNA. Next thing you know, you're infected. And they were working on this in the late 1990s. So here's the abstract. Coronaviruses generally have a narrow host range, infecting one or just a few species using targeted RNA recombination. We constructed a mutant of the coronavirus mouse hepatitis virus in which the ectodomain of the spike glycoprotein was replaced with a highly divergent ectodomain of the S protein of the feline infectious peritonitis virus. So there you got, they're taking in, they're what are they taking the feline aspect away and they're making it so it can infect mice now. And they're using recombinant techniques and test tubes to do this as well to find the perfect match. The resulting in creating chimeric viruses. The resulting chimeric virus, designated female mouse hepatitis virus, acquired the ability to infect feline cells and simultaneously lost the ability to infect murine cells in tissue culture. This reciprocal switch of the species specificity strongly supports the notion that coronavirus host cell range is determined primarily at the level of interactions between the S protein and the virus receptor. The isolation of female mouse hepatitis virus allowed the localization of the region responsible for S protein incorporation into virons to the carboxy blah blah blah. This establishes a basis for further definition of elements involved in virin assembly. In addition, FMHV is potentially the ideal recipient virus for carrying out reverse genetics of MHV by targeted RNA recombination, since it presents the possibility of selecting recombinants, no matter how defective, that have regained the ability to replicate in murine cells. So they're saying it's a good one for these reverse genetics of SARS because, you know, it can infect the new hosts. Maybe they wanted to infect humans, so they'll add the human spike protein on there. And then, but simultaneously, it loses the ability to infect the previous host. So that's why this is going to be good for future scientific work. I'm posting this in the chat for you guys. I know we got my complainers that go, hey, post your work in the chat, Daz. so I don't have to go searching on the internet for it myself. So I got another one here. Welcome to Caveman Science. If you never watched a Caveman Science before, ladies and gentlemen, Caveman stumbling through scientific research. It doesn't get better than that. So this took place in the late 90s. It was officially documented in February 2000. Assembly of spikes into coronavirus particles is mediated by carboxy terminal domain of the spike protein. So you have them manipulating the host cell's species barrier, and you have them here manipulating the spikes of coronaviruses. Now they're like creating new coronaviruses that never existed in nature. Now this is the pre-original SARS outbreak in 2000, late 2002. I believe it happened in Guangdong, China, in November 2002 was the original outbreak. So we have like new coronaviruses, not yet labeled SARS, being researched and being created in a laboratory setting before the original outbreak. So I'm going to read this abstract real quick. I think assembly of spikes into coronavirus particles is mediated by the carboxy terminal domain of the spike protein. The type 1 glycoprotein S of coronavirus trimers, of which constitute the typical viral spikes, is assembled into virins through Actually, I don't think I'm going to read this. Read it yourself if you want to. You get the point. They're assembling the spikes. Here's the next one. I'll share it in the chat. Sorry, I didn't want to get too sciencey. Sometimes Andy stumbles when it gets a little too sciencey. So this was, I guess, before I should have read this one first. Coronavirus particle assembly, primary structure requirements of the membrane protein. So they were pretty much trying to build a coronavirus from scratch, it seems like, in a laboratory setting, while simultaneously coming up with different recombinants and different coronaviruses that never before existed in nature. Now, there later on, a bunch of synthetic biology is being developed, they incorporate that technology as well, and I think that's when you get the SARS-CoV-2 starts coming into play, is when they start incorporating synthetic biology. But yeah, I got what are the three I just showed you? The coronavirus particle assembly, primary structure requirements of the membrane protein. So this is like your outer, like the gel on the outside of the cell, and then you got the spike on, which I just read before, that assembly of spikes into coronavirus particles. So they built the membrane, they added the spikes, and then they started adding different host cell species after that. That's pretty much what I'm getting to here. So let me show this in order. This is them in 1998, coronavirus particle assembly. They're assembling your basic coronavirus cell. Trust me, I know I'm stumbling through this. It's caveman science. The next one, a little bit later on, assembly of spikes into coronavirus particles is mediated by the carboxy terminal domain of the spike protein. So you got your gel on the outside of your cell. Now they're adding the spikes to the outside. And then the next one, retargeting of coronavirus by substitution of the spike glycoprotein ectodomain crossing the host cell species barrier. So this is them adding the, like, if you want to infect mice, if you want to infect a cat, civets, a human, this is them starting the work on that process. Now you guys are asking me, like, what does Ralph Baric in North Carolina have to do with this? I've done a lot of work previously that they were involved, but they were involved in the original stuff as well. So here you got Ralph Baric and his lab partner Amy Sims, who invented remdesivir together with Timothy Sheehan in North Carolina. Development of mouse hepatitis virus and SARS-CoV infectious cDNA clone DNA construct. So they were using this mouse hepatitis virus and SARS-CoV infectious clone products, and this is in 2005, and I believe this work started in 2003. So they're using that like genetically engineered mouse hepatitis virus cell, and they were trying to add some SARS-CoV infectious clone spikes and mRNAs to the virus to create vaccines in their North Carolina laboratory. And then here you have later on Ralph Baric and his team. They use reverse genetics with a full-length infectious DNA, cloned DNA of severe acute respiratory syndrome coronavirus. English, please, Andy. Hey, Joe Biden. Why are you reading scientific research papers on camera, Joe Biden? That is what I feel like sometimes. It's like, hey, I'm not the best. If Joe Biden can make it as president, this stumbling, mumbling guy can make it as well. So, reverse genetics with a full-length infectious clone DNA of SARS coronavirus. And this was published in 2003. So the work was started before this. So they were reverse engineering SARS, the original one, like early 2000s. So abstract. A previously undescribed coronavirus is the etiologic agent responsible for severe acute respiratory syndrome. Using a panel of contiguous clone DNAs that span the entire genome, we have assembled a full-length clone DNA of the SARS-CoV Urbani strain and have rescued molecularly cloned SARS viruses, infectious clone SARS-CoV, that contain the expected marker mutations inserted into the component clones. Recombinant viruses replicated as efficiently as WT virus, and both were inhibited by treatment with cysteine proteinase inhibitor. I'm not going to get into all that, but there you have it. They created a fully infectious clone model of SARS. So one other thing, I'm going to kind of get out of the actual. I'm not going to fully get out, but I'm just going to read a couple Ralph Baric things just to showcase that he is 100% like the man behind all this. So here's the Federal Register from 2004, and it has to relate to the work of on SARS, work and research on SARS. From the Department of Health and Human Services, Centers for Disease Control and Prevention. Role of the environment in the transmission of SARS-CoV. Notice of intent to fund single eligibility award. So it's like, who's going to get the work for the research of SARS? So B, eligible applicant. You notice they don't say applicants. There's only one applicant that is eligible. Now, this is in the early 2000s. Assistance will be provided only to the University of North Carolina. So North Carolina is the only one that's receiving SARS-funded research from the HHS and CDC in 2004. And this came out. Experience of UNC staff. It is in the best. Sorry, my brightness keeps going down. It is in the best interests of CDC to utilize the expertise of Dr. Sabzi Weber, and Rutella from the University of North Carolina, who have combined for over 80 years of experience in microbial inactivation studies using a wide range of microorganisms, including a wide range of viruses. The UNC BSL-3 laboratory is shared with Dr. Ralph Baric, who has spent the last 20 years studying how coronaviruses are transmitted among species. Dr. Barrett currently has research support from the NIH for a variety of research projects involving SARS. Thus, UNC has the unique opportunity to collaborate with Dr. Barrett, one of the world's experts in coronaviruses. Lastly, UNC has a cadre of researchers that are well trained in microbial inactivation studies and have published several hundred papers on this subject. Urgency of the need to address the SARS-CoV research questions. The emergence of SARS produced an international health emergency in late winter and in the early spring in 2003. By early July, there were an estimated 8,439 probable cases and 812 deaths from SARS. Immediate availability of BSL-3 laboratory. The University of North Carolina BSL-3 laboratory is now available to conduct the research. This will enable a timely response to research questions regarding how long infectious virus can persist on common hospital environmental surfaces, wastewater, etc., or the role of personal protective equipment for protecting healthcare workers. While other institutions may have BSL-3 capability, the facilities are usually restricted to use with a limited number of infectious agents. For example, a facility conducting work on Mycobacterium tuberculosis would not use the same BSL-3 facility for working with coronaviruses, since disinfection schemes would be different and the necessity for cell culture materials and unique pieces of equipment would likely require remodeling. A facility such as that at UNC that is already equipped to work with coronaviruses saves considerable expense in retooling a BSL-3 to work with this virus. Now they get into some of the grants and some of the work that will be done. So on this next one, I'm almost done here, ladies and gentlemen. On this next one, this is like Ralph Barrick's actual curriculum that he was working on, his research curriculum that he's working on at UNC when it comes to coronaviruses and SARS and reverse engineering them and creating them in his lab. And so here's this paper. I make sure you save it and document it for all you researchers out there. It's an important one. I hope these are working. An important one. It just gets into all his work and how it's funded by the NIH pretty much. And if you go through it, I'm gonna scroll. See right here, he's working on macaque model and gene expression profiling SARS. I'm gonna get into some of his completed projects here real quick. Let me find my spot. So this is Ralph Baric working on coronavirus, and this is like from the get-go. This is his completed work that was funded by our government. So here we go. Studies into the mechanisms of this mouse hepatitis virus, and this was in 86 to 90. Coronavirus induced myocarditis in rabbits. That's from 1987 to 1990. Now remember, he's also working on like host cell. Eventually, they'll start working on like host cell species, trying to cross that to make it into humans. So coronavirus induced rabbit cardiomyopathy. That was from 89 to 94. Coronavirus induced myocarditis in rabbits, 86 to 87. Incidence of enteric rotavirus, adenovirus, and coronavirus among migrant farm workers, 1987 to 88. Is it coming over the border, ladies and gentlemen? Small instrument program, coronavirus-induced myocarditis and dilated cardiomyopathy, 90 to 93. Development of PCR techniques. Gene probes to analyze for waterborne microorganisms and viruses. Detection of human and non-human fecal indicators in shellfish. Sorry, I'm just reading now. You get some, there's more coronavirus stuff. Development of a molecular method to detect infected viruses. Reverse genetics with a coronavirus infectious clone DNA construct from 2001 to 2005. So that's a big one. Wouldn't you say they were the NIH funded pretty much creating new coronaviruses in a lab that didn't exist in nature? Oh, and that was, what year did that research start? 2001. That's the 4-1. The original coronavirus SARS-CoV-1 broke out in, what was it, 11/1 2002, and Ralph Baric and the team were working on it in 4/1 2001. Remodeling the SARS coronavirus genome regulatory network. That's something he was working on. They remodeled and rewired the coronavirus gene, and that started in 2004. SARS reverse genetics. You guys get the point. If there's a scientist out there that created SARS in a laboratory, where it was at least on the original team, it was Ralph Baric. And it happened in North Carolina. Here's a patent. Everyone loves their patents. Methods for producing recombinant coronaviruses. United States patents. Filed in April 19th, 2002. So before the first coronavirus ever was on, the first SARS-CoV was ever on the scene in China. Guess who filed the patent for producing coronaviruses that never existed in nature? Ralph Baric in the University of Chapel Hill, North Carolina. Dun, dun, dun. They filed it in 2002, April 19th, 2002. Got approved October 9th. That doesn't mean they didn't file it, though. You know, you file the patent for making the recombinants because you whoever makes the vaccine, you want your hand in the pie too. My germs. What's that movie? So that's my show for today. I'm sorry for stumbling through a lot of my scientific research. You know, it's been a while. I'm warming up again. I'm getting back into it. My little hospital stay, I was literally like a zombie for like 4 days in the hospital. I haven't even been out a week. I've only been out like 4 days. But I just like sat there, stared at a wall. I don't think I ate one thing for 4 days. I was not hungry at all. They were dousing me with drugs, so I'm still on drugs. If I seem a little low energy, if I seem a little Jeb Bush like, a little low energy, it's definitely because they have me on like a bunch of heart medications. It affects my vision a little bit, to tell you the truth, to be honest. I'm sitting here reading and stuff. It's like, it's hard to read. So forgive me when it comes to stumbling through reading some of the scientific research. I'm normally not that bad, but I will get better. I will improve. I will get back to normal sooner or later. George Webb is joining me on Wednesday I believe he's jumping on at 7:30. I'm going to start my show at 7, do a little George Webb preamble and some of the things we're going to be talking about. And then he'll be jumping on at 7:30 and we'll be breaking down and actually we'll be catching up. It's been a while since he came on the show last Wednesday. I think it was last Wednesday. We had a pretty good show. Then the next day I was in the hospital. So hopefully it doesn't happen again after this upcoming show. But he's joining Wednesday. I got a couple other people that I will be interviewing and on the show here pretty soon that I'm looking forward to. But other than that, make sure you guys get your workout in. 25 push-ups, 50 sit-ups. I think that's what we're up to now. Make sure you get your exercise in, ladies and gentlemen. We don't want no fatties at the beach in the lake this summer. If I see you out and about, if you're a fan of mine and I see you at the lake, if I see you at the beach, if I see you at the park with your shirt off and I see a little mushroom top hanging over your belt there, I'm going to come up and give you a little red belly. I'll be like, that one. It's for not staying disciplined. All right, ladies and gentlemen, have a good evening. I don't even know if anything I said tonight made sense. It was one of those shows where it's like I feel like I'm getting back into the groove. I have no idea what I even like talked about at certain points, but all I know is the original SARS was genetically engineered, and I have the documents to prove it. If you want them sent to yourself, just email me at livewithandy@gmail.com. Be like, send me those documents, Andy, and I'll put them in your mailbox and let you deal with them. I don't want the responsibility myself. 50 sit-ups in exercise snack. I know. I'm not even asking that much, Deborah. That's just like you're barely even breaking a sweat. It's like a starting point. 25 push-ups and 50 sit-ups. Just basic. And then in like 3 weeks, we'll be doing 50 push-ups and 100 sit-ups and going for a 2-mile walk. Then by the end of the summer, you're going to be looking like Arnold Schwarzenegger out there. Toledo loves. That was a terrible Arnold Schwarzenegger impersonation. All right. I'm going to let you guys go for the night. Thank you for tuning in once again, George Webb. Thank you for joining on Wednesday at 7:30. Follow me on Twitter at LivewithAndy. Follow me on YouTube. Just put at Andy Dybala or at Andy Dybala clone into YouTube, and my little videos will pop up. I do a lot of YouTube shorts, and I post some of my research on Twitter. So make sure you follow me on those in between shows so you can catch up on what's going on. Have a good night, people. Love you show.